Okay, I am doing a follow-up lesson on neurons because one of our previous lessons was about neurons and I feel like there was confusion. So I want to do a follow-up. So as you can see right here, here is a structure of a neuron as we typically see it. This is not the only structure for a neuron, but is a very common one. So let's see. We have a cell body with a nucleus. It doesn't just have the nucleus, it has the cell organelles, so the mitochondria, the ribosomes, all those things that help the cell to function, the endoplasmic reticulum, all that stuff would be found in the cell body. And then from the cell body comes these extensions in this diagram, the dendrites, and they're the receivers. They pick up signals from other neurons. So think of them like TV antennas. Okay, they're all branched out, pulling it in, pulling it in, pulling in the signals, picking them up. Okay, then from there in the diagram, from the cell body with the dendrites, the little extenders picking up signals, we have a long axon, and that's usually the long, thin part of the neuron, although if the neuron is very small, the axon may also be very small. The axon ends with these axon terminals. At the end, they look like little buttons, and that's where some major action happens, which I'll talk about. Now, a lot of axons are covered with a substance called myelin. So there's this myelin sheath. And you could see this little pink spot on there, and that is the Schwann cell. The Schwann cell is what produces the myelin. And what is the point of it? Well, it insulates the axon and helps the signal get transmitted a lot faster. But you can see it is not a continuous sheath. There are gaps. And at each space where the axon is like exposed, not covered with myelin, it is called a node of Ranvier. So what happens is, and I'll show this in another illustration later, is the signal kind of skips across most of the axon and can just have an action potential occur at the node, and does not need to have that happen along the whole length of the axon when there's myelin. When there's not myelin, it's different. So we'll see that in a later slide. Here is a close-up of a synapse. So what is a synapse? A synapse is the space between two neurons. So two neurons do not touch. So they get infinitely close to each other. So imagine the synapse as like your sibling, like, I'm not touching you, like, don't touch me. And they're like, I'm not touching you, but they put their finger like this close. So they're like so close, I'm not touching you, I'm not touching you. Well, that's how neurons are. They get infinitely close, but they're not actually touching each other. There's a space between them called the synapse. So if the neurons aren't touching, they have to have another way to get the signal across that synapse. There's the space. So what happens is they use neurotransmitters. So here's the neuron. It came down the axon. Here is the button, right? This is the axon terminal. And the axon terminal has these vesicles that contain neurotransmitters in them. And when the signal gets all the way to the end, the vesicle is released, the neurotransmitters are released into the synapse, and they cross to the other cell. Now, the neurotransmitter has to match up with a specific receptor. So if that is acetylcholine, it has to match with the acetylcholine receptor. If that's dopamine, it has to match with the dopamine receptor. If that's serotonin, it has to match with the serotonin receptor. Okay, so it's very specific. And there are things that can affect what happens. So things that can go wrong. Well, there might not be enough vesicles with, say, dopamine in them. Well, that's a problem. Okay. There may not be enough receptors, also a problem. There might be too many receptors, also a problem. There might be something blocking the receptors, also a problem. And you could see in this GIF, the cell, once the dopamine is used, signals the receptors, it is recycled. So it reuptakes that dopamine to use it again. So if there's an issue with the reuptake, that also creates a problem. So there are a lot of spots where there can be issues, all right? But this is what happens in a synapse. The cell, this is the end of the axon. This is the axon terminal. It releases the neurotransmitter into the synapse. Receptors on the dendrites of the next neuron are what allows the signal to be passed on. Okay, so there are three main types of neurons. Let's talk about these. We have sensory neuron, and you could see from this illustration of the sensory neuron, do you see how the cell body is in a different place? Okay, so again, all neurons do not look exactly the same. So we have sensory neurons, which are afferent, A for afferent, like affect. And then we have motor neurons, efferent, like effect. And then we have interneurons, they're the connectors. Okay, so what happens is a receptor is signaled in a sensory neuron. It sends the signal, it picks up the signal, the dendrite, sends it all the way down the axon, okay, and then it is close to the dendrites on an interneuron, picks up the signal, sends the signal all the way down to the dendrites of a motor neuron, and then that is what is going to cause a response. Okay, now you can see right here the interneuron does not have myelin around it in this illustration. So primarily because it's a very small neuron, so myelin may not be needed. So not all neurons have myelin, but a lot of them do. So here's the difference when we have myelin and when we don't. So what an action potential is a change in the charge of a neuron. And it's ions cross the cell membrane to cause that change in charge. So if there's no myelin, so this is the first right here. So if there's no myelin, it's going to go down the whole length of that axon, switching from a negative inside to positive inside, and then switching back. Okay, and it's going to go like that all the way down the neuron. And that is right here in this illustration. You can see that's a relatively slow process. In some cells, the axon is very long, so that's a long time in order to have those charges moving. Okay, you can see here though what happens with myelin. Do you see how the signal just skips to the node? Imagine crawling. That's no myelin, and skipping with myelin. You literally, when you're skipping, your foot is only touching for a very brief moment, and then it's back up again, and then it's down, and then only for a brief moment. So the signal, when there's myelin, can kind of skip down the axon, and the signal gets transmitted sometimes hundreds of times faster. Okay, so that is very important for getting nerve signals transmitted quickly. All right, so now probably the toughest part of neurons is this whole thing, this action potential. So this is what's happening. Remember, it's negative in the cell, it's positive outside, and then there's it switches, and then it switches back. So like, what the heck is happening? So I'm going to try to break it down into the easiest way I can. So at resting, neurons at rest, there's a lot of sodium outside the cell, and there's a lot of potassium in the cell. Now, naturally, some of the sodium leaks in and some of the potassium leaks out. That's just a natural process because there's so much sodium outside, so some of it kind of sneaks in. And there's so much potassium inside that some of it sneaks out. But we aren't ready for it. So there has to be a pump to fix it. So a sodium-potassium pump is like, hey, sodium, stay outside. Hey, potassium, get back inside. So it switches it. So it keeps the concentration relatively the same. More sodium outside, more potassium inside. And the inside of the neuron is negative. And how I remember this is DNA has a negative charge. If you think about electrophoresis in biology and how DNA is able to be pushed along, it's because DNA has a negative charge. So I always remember that because DNA is negative, it's inside the cell. So the inside of the neuron has a negative charge. Now, what happens is the neuron starts getting signal. So it's getting some receptors that have been triggered by neurotransmitters, okay? So what happens then is some of the sodium channels, gates, pathways, passages, however you want to think about it, they open. So sodium starts coming in slowly. And you can see in this illustration, you can see how it starts to move upward slowly. And there's this magic number right here, threshold. As soon as it reaches threshold, you could see all of a sudden it jumps up super fast. So it's like sodium is, some of the gates are opening, okay? Imagine we're at a concert and there are 20 doors to get into the concert. and it starts getting close to time, so they open 2 doors. So people are getting in slowly. Then all of a sudden, the opening time hits, and they open all of the doors. That's what happens as soon as we reach threshold. Then all of a sudden, sodium is like whoosh and rushes into the cell. And that creates that depolarization, whoosh, sodium rushing into the cell so fast, okay? But at the same time, then the cell is like, oh my goodness, now it is so positive inside, and it is supposed to be negative inside. So the fastest way to fix that, okay, because the inside just became positive, you can see the chart up here, is in this process of repolarization, the potassium gates open, and we let the potassium leave in a hurry, and it makes the inside negative again. But the cell is so ambitious in this process that usually it lets too much potassium out, and you can see right here this little extra dip. That's called hyperpolarization, like it got too negative. The cell was like, whoa, tried to fix it too much. So let me go through it again. Resting, some sodium is leaking in, some potassium is leaking out, but I'm using a pump to kind of keep it stabilized. The neuron starts getting signaled. Couple sodium doors open, sodium starts coming in slow, reach threshold, bam, the sodium gates open and sodium floods in. And then that is depolarization. Inside becomes positive. Cell panics. Oh my gosh, fix this fast. Opens the potassium channels. Potassium rushes out in a hurry, and so much potassium rushes out that the cell becomes extra negative. Hyperpolarization. Now you think that would be the end, right? Negative. Why? Because the sodium and potassium are in the wrong spot. So the neuron resets. So we use a sodium potassium pump to push sodium back out and push and pull potassium back in and reset the neuron so it can be ready for the next signal. Okay, so I hope that helped explain some of those difficult things because there seems to be a lot of misunderstanding, and I find this action potential fascinating. So this is an action potential, and hopefully it helps make a little bit more sense to you. All right.